Hello students, welcome to LEELA'S tutorial. In last class we have discussed about plant tissue culture where you are culturing cells, tissues or organs in vitro to produce a complete plantlet. Right? So for this in vitro process we require certain nutrients which are necessary for the plants to grow. Right? So let us see today nutritional requirements for plant tissue culture. So these are the basic requirements. So the first requirement is essential metals and minerals. So here you have, it is classified into macro elements and the micro elements. So what do you mean by macro elements? These are the elements which are required in higher concentrations, right? And hence they are referred as macro elements such as nitrogen, sulfur, phosphorus, potassium, magnesium, calcium, etc. Then you have the micro elements. These are the metals which are required in very low concentrations, right? The minimum amount of these elements is sufficient for the plants or the cells to grow. That is iron, boron, zinc, molybdenum, manganese, chlorine, etc. Right? So here, apart from these basic essential metals or the elements, right, you have to supply the energy source. We know that the basic energy source are your carbohydrates. For example, glucose and sucrose. These are readily available. They are easily absorbable by the cell, right? And that can be easily transported through the tissues, right? So in between the cells or the tissues, this glucose or the sucrose can be transported very easily. So and also they are mainly required for the increased growth rate. Okay, apart from this energy source, we are going to add the growth regulators. These growth regulators are also referred as growth hormones, right? So in plant tissue culture media, apart from your basic nutrients, that is macro and micro elements, here these growth regulators act as media additives, right? So we are providing additional nutrition, or addition, you are providing certain hormones in order to initiate the required growth, right? Required growth in the sense, if you want only the root to get elongated, you give auxins. The shoot, you give cytokinins. If you want the stem to increase, you give gibberellins. And abscisic acid, it is a negative growth hormone, whereas auxins, cytokinins and gibberellins are positive growth hormones which help in increasing the shoot and the root system. So each one of them we'll discuss in detail in next class. Next you have the proteins which can be provided in terms of peptides or amino acids like asparagine, right? So here all these amino acids or the proteins act as nitrogen source. So these are readily available nitrogen, or these can be absorbed easily when compared to your inorganic nitrogen, right? Again you have vitamins. So vitamins, these also help in increasing the growth or increasing the rate of growth. They fasten the growth. So what are vitamins used? It is thiamine, nicotinic acid, that is B1 vitamin, B3 vitamin, then you have pyridoxine, it is B6, then myoinositol, folic acid, etc. So these vitamins enhance the growth rate. Then you have antibiotics, that is streptomycin and kanamycin. So here these antibiotics must be added to your media in order to avoid the microbial contamination, right? In order to avoid bacterial or the fungal contamination in the plant tissue culture, we make use of certain antibiotics. Then you have the buffering agents. So in case of plant tissue culture, we have already talked about the suspension culture where we go for the production of secondary metabolites. So in that case, as the cell is growing or as the cell is producing the secondary metabolites, there may be change in the pH, right? So in that case, you can maintain the pH by using this buffering agents such as phosphate or the acetate buffers can be used. Then you have the other supplements such as coconut milk or tomato juice. So here these other supplements in terms of, see, all these things we call them as chemically defined synthetic media. That means these all chemically defined synthetic media means all the components, right, and all the compounds present in the media are known. We know what is the component you are going to add, right? You are going to weigh them accurately and then add it. That means you know the compound and also its composition. But here in this case, in other supplements, it is chemically or it is natural undefined media supplement. That means these are the natural products and you don't know what is the concentration of certain products present in them. You are adding tomato juice. You don't know how much of lycopene present in that tomato juice, right? And here the coconut milk. In the same way, you know the compound. What is the compound present in that? But you don't know the exact composition of that particular compound, right? So these are undefined natural products which you are adding as an additional supplements. Then you have the gelling agents. So in last class we have discussed about semi-solid media and also the liquid media, right? Liquid media is also referred as suspension cultures. So here if it is a suspension culture, right, you want a liquid media, then you will be adding all the basic components with the supplements, right? It can be defined or undefined media. You are adding everything. Then as you want cell suspension culture process, you are not going to add any gelling agent. In case if you want a semi-solid media over which you want to produce a callus or inoculate the explant, then in that case, after adding all these substances, you are going to add the gelling agent such as agar or agarose. Then only you are going to keep it for the autoclaving, that is sterilization of the media. So here you are going to sterilize all the compounds. You are going to dissolve this agar or the agarose, whatever you are using, you are going to dissolve them, then keep it for autoclaving. So what happens if there is any contamination in the media during preparation? Everything, all the microbial contamination or the fungal contamination can be eradicated by autoclaving or sterilization process. Condition, here you have one condition. Here you have the coconut milk, tomato juice, orange juice or banana juice. So here all these components are heat labile compounds, right? So here if in case if you are adding any heat labile compound, you are not going to sterilize them by using autoclaving, right? If these compounds are heat stable, you are going to add all heat stable compounds and go for autoclaving, right, for sterilization. After they get cool, right, let them get cool, then only you are going to add your heat labile compounds, right? There may be again contamination due to the heat labile compounds, that is heat sensitive compounds. What do you mean by heat sensitive compounds? That means these compounds consists of certain proteins which may get denatured due to high temperatures, right, which may get denatured due to heat. So what you are going to do, you have to sterilize them. So instead of using heat sterilization process or the steam sterilization process, you may use filter sterilization. Filter sterilization we'll discuss in detail, right? So remember that only the heat stable compounds will be sterilized in autoclave, and after cooling you are going to add heat labile compounds which are sterilized by using filter sterilization apparatus, right? After adding all these, you are allowing it to set, right? If you want a semi-solid media, you are going to use the gelling agents. If it is a suspension culture or the liquid media, you are avoiding this use of gelling agents, that is agar or the agarose, right? Based on the requirement, you are going to prepare the liquid media or the semi-solid media. Okay, so here these are the basic components which are present. Okay, based on the composition of the components, they have named as different types of media. So if you come to the types of media, mostly we make use of MS media, that is Murashig and Skoog media. Then you have B5 media, that is Gambox media. Then you have EME media, that is Eagle's minimal essential media. Right? See here, in all these cases, the basic components may be same, but their composition will differ. Okay? So based on the explant used or the type of culture, whether it is a suspension or a semi-solid culture, right? You are going to select the media and proceed for the plant tissue culture process. Is that clear? If you have any doubts, put it in comments.